Yeah, so our final speaker is Vicky Mack, and she is a research assistant at Columbia University in the lab of Aaron L. Barnhart. And her talk is on exploring the role of the endoplasmic reticulum in regulating activity-driven intracellular calcium signaling in Drosophila elementary motion detection. So whenever you're ready.
Sorry, that was a mouthful, I know. Okay. Oh, my bad. Okay, so in the Barnhart lab, our fundamental purpose is to examine the cell biology of neurons. This includes looking at how intracellular activity affects single cell processing and overall circuit function in vivo. If we can understand neuronal processing at the cellular level, imagine the large-scale effects we can see on an animal's brain and behavior if we alter regulatory mechanisms within the neuron. For this project, I'm studying how calcium handling in the endoplasmic reticulum, or the ER, affects neuronal function in the elementary motion detection circuit of the Drosophila. One of the major stores for calcium in the neuron is the ER. The ER stretches throughout the entire neuron, and calcium can flow into and out of the organelle within the cell. Calcium is an important second messenger in neurotransmission, since it initiates synaptic vesicle fusion and triggers neurotransmitter release from vesicles. We're focused on understanding how calcium handling in the ER can ultimately shape cytosolic calcium signals and consequently shape neurotransmission, and how ER calcium handling can affect the function of not only that single neuron, but also within an entire circuit. Although the molecular mechanisms driving calcium handling in the ER has been studied in cell cultures and slides preparations, there have been lack of studies showing how this process works in vivo. So, the Drosophila visual system provides a highly stereotyped cellular architecture, since its visual circuit is very well defined. The Drosophila elementary motion detection circuit operates by extracting and comparing luminance signals from adjacent points of space at two different points in time. Elementary motion detection is comprised of different types of neurons that detect local motion visual cues. On the left, photoreceptors project onto primary lamina neurons. Lamina neurons split signals into the on and off visual pathways. In the on pathways, secondary medullary neurons depolarize in response to light increments or lights on, and hyperpolarize to light decrements. In the off pathway, these neurons respond in reverse. They depolarize in response to light decrements and hyperpolarize to light increments. Medullary neurons respond differently to visual stimuli, having different response kinetics. In the right figure, the two curves represent the responses of two different neurons. Notice how one has a faster signal than the other curve. These different timing responses determine whether a signal is either suppressed or enhanced. These third order neurons depend on this temporal filtering to generate direction selective motion signals. These signals are pulled and processed by projection neurons traveling to the central brain. This allows flies to detect different optic flow patterns, such as motion in different directions. We think that calcium handling in the ER contributes to the timing response differences of medullary neurons and affect the temporal filtering of these cells. For this project, my experiments are focused on the Mi1 neuron, which belongs to the on pathway and projects onto direction selective T4 neurons. So for my experimental plan, I'm conducting 3 broad experiments on ER-mediated calcium handling in the Mi1 neuron. First, we want to know whether there are responses in the ER to visual stimuli in the flies. So first we need to correlate ER and cytosolic calcium signals across different compartments in Mi1 neurons. The second experiment is concerning how the ER and cytosolic calcium responses, how they respond at all to molecular perturbations to the ER. And lastly, to determine how The ER contributes to overall circuit function. We want to measure downstream effects within the cytosol of downstream neurons as we perturb the ER in Mi1 neurons. So for our experimental setup, the fly's head is dissected first, while the entire fly is mounted to the plastic holder. The fly is then placed to face the stimulus screen, which will display 2-second light flashes periodically. On the right is a recording of Mi1 cytosolic calcium signals to light flash stimuli. As you can see, it just responded very brightly to light flash on, and then it decreased in brightness to light flashes off. Okay, so for our first experimental plan, we want to determine whether the ER does respond to visual stimuli at all. So the Mi1 neurons have 3 different compartments. They have dendrites in layers M1 and M5 of the medulla, and they have axons in layers M9 and 10. And so usually in vivo two-photon microscopy imaging, we measure cytosolic and ER calcium responses across a period of 4 seconds. As we measure the different fluorescent intensities till light flashes on, indicated in a lighter green or lighter purple color. We also measured responses to light flashing off in darker green, darker purple colors. We could see across the cytosol that calcium responses increase. They have very fast responses, and they also have very sustained responses at the stimulus onset when the light flashes on across all 3 different compartments. So this shows a homogeneity across different compartments. This is a predictable response from what we know about on-pathway visual neurons. But in the ER, this is the first time that we're seeing that the ER does respond to physiological stimuli, but they also respond heterogeneously across all of these compartments. So in layers M1, there is fast transient calcium uptake when the light flashes on. However, in dendrites of layer M5, there is a slow, sustained calcium release in the ER to light flashing on. And in the axon terminals, we see that there is a fast yet sustained calcium uptake at the stimulus onset. So first, we show that the ER does respond to physiological stimuli. Second, these responses are heterogeneous, meaning that this suggests that the ER has different functional needs across different compartments within a single cell type. So for our,
all right, yes. Just wanted to butt in real quick. It's been about 8 minutes. I just want to let you know, you might want to wrap up soon.
Oh, sorry. So for the second experiment, we wanted to measure ER and cytosolic calcium responses when we perturb the ER. And so there are different molecular mechanisms that drive ER calcium dynamics. When a signaling ligand attaches itself to a G-coupled protein receptor, this generates IP3. Both IP3 and calcium together activate IP3 receptors located on the ER membrane. These IP3 receptors, when activated, allow for calcium release from the ER into the cytosol. The only way that the ER replenishes its calcium levels is through sarcoplasmic calcium ATPases. So when we knock down, when we knock down IP3R, we wanted to see if this alters ER Ca2 signals in Mi1. So in layers M1, there is not really too large of a discernible differences between both the control and the IP3R RNAi knockdown. In layers M5, however, we can see that calcium uptake and calcium release are both diminished. And in axon terminals for layers M9 and 10, we see that calcium uptake is more sustained in the IP3R RNAi knockdown. And calcium release as well is has been less sustained. So we can see that the IP3R RNAi has subtle effects across all different compartments within a single cell type. But what does that, how does that affect the cytosolic calcium responses? We can see that across all different compartments that cytosolic calcium responses are more sustained when you knock down IP3R receptors. So from these results, we considered that perhaps the ER has a compensatory mechanism in which when there is a deficiency in IP3R activity, then the ER, through some means, is able to decrease circuit activity as well. So there must be a mechanism within the ER that is able to detect calcium levels within the organelle to determine and to modulate both uptake and both release of calcium within the organelle.
I think we're kind of out of time at this point. I think we should probably leave some time for questions. If you could wrap up real quick, that would be great. Just like a quick conclusion.
Yes. After gathering all of these results, we want to later on see how the ER plays a role in mediating calcium signaling downstream in overall circuit function. By understanding how ER plays a role in single cell processing and large scale circuit function, we are able to understand how intracellular activity can have an overall broad scale effects on how we are able to process information from our surroundings and make decisions and think decisions on how we choose to behave to these sensory stimuli. If you have any questions, feel free to let me know, and I'll do my best to answer them. I'd like to thank the Barnhart Lab for supporting me throughout this research project.
Awesome, fantastic talk. Thanks, Vicky. So we do have a question in the Q&A. It says, nice work. Sorry if I missed this, but how can you differentiate between the cytosolic and endoplasmic reticulum Ca2 response? Is your imaging resolution high enough to differentiate those different intracellular compartments?
So it's not really a matter of imaging resolution, actually. It's more of that we use different types of calcium indicators for both the ER, and the cytosol. So let me see. So the ER and the cytosol have different calcium concentration levels. The ER has a cytosolic the ER has a calcium concentration that is 3 orders of magnitude higher than that of the cytosol. So using that, the Ryan lab has created a GCamp calcium indicator for the ER that has a low binding affinity, such that we are able to view 2 different calcium indicators for both the cytosol and the ER in a cell type specific manner.
Great. Awesome. Well, just want to thank all 3 of our speakers.